Hi, my name is Tom Manning and I'm a professor at Valdosta State University in the chemistry department. I'm here to tell you about my passion. My passion is science education and in particularly innovative methods. I like to call what I do teaching at the peer review level. That means when students are given a project that it potentially can have some impact on an important topic and it can be published in a journal or receive a U.S. patent. So I'd like to tell you the story of one of them, the Bryostatin story, but I should point out that that is one of 5 or 6 projects we have going now. We have others going in the development of new cancer drugs. We've had 20 cancer drugs enter preclinical trials. We have drug-resistant tuberculosis is one of our projects. We've developed an antibiotic that works against that. We have done work with an Alzheimer's drug that I'll talk about in a second. We've also done work with coral and oyster restoration. So let me tell you about Bryostatin and how undergraduates can produce very competitive research results using just a simpler methodology. Okay, Bryostatin goes back to John Steinbeck and his books Cannery Row and Sea of Cortez about a collector named Doc. Doc was a real man that lived on the coast of California and collected marine specimens and sold them across the country. In the 1960s, when Steinbeck was growing old, there was a young man in a Florida panhandle named Jack Rudlow, and Steinbeck encouraged Rudlow to open a lab that eventually was called Gulf Specimen Marine Lab. Jack participated in a National Cancer Institute collection of over 300 marine species that was searching for the first cancer drug to come out of the ocean. Out of that, an obscure marine creature called Bugula narentina stood out against the leukemia cell line. Our role came in perhaps almost 20 years ago when we went to Mr. Rudlow to look at Bugula narentina and some other creatures. In that large-scale survey of the 300 species they collected, which ranged from sharks to sand dollars, there were about 30 that stood out that had some activity for leukemia. We collected about 25 of those, and we did an extraction and we found that Bryostatin was in 25 of them at very low levels. Okay, so what we did from that point on was to try to synthesize it. Currently, Bryostatin is used for Alzheimer's. It's in clinical trials. Has strong efficacy against many types of cancer, and it attacks HIV reservoirs that retrovirals do not attack. But its synthesis is still considered extremely difficult, and the drug now costs about $15 million a gram. We wanted to develop a method that would be much cheaper. So working with undergraduates, our problem to solve was to make that drug much cheaper than the elite universities could do and the large pharmaceuticals. While many of those organizations were concentrating on a technology-driven, lab-based synthesis, we went back to the ocean. We identified an area where that bacteria that made the drug lived, and it actually lived within Bugula. It was symbiotic, and then we worked on a method we call pharmaceutical aquaculture. All the steps that we do are relatively low technology, inexpensive, and it results in an economical product. We've had one form of the drug, an iron bryostatin complex, enter preclinical trials at the National Cancer Institute, and now we're isolating other forms of it. Do students get excited about going to the beach to collect samples? Yes, they do. Can they do everything in the lab that we ask of them as undergraduates that only have a few hours here or a few hours there free? Yes, they can. Is our method economical and green? Yes, it is. So this is what I'd like to talk about in my TED Talk, is my passion with attacking complex problems and then trying to come up with a much simpler system. Thank you very much.